Morning, everyone. Sorry, brief introduction. Are we running ahead of schedule? Yeah, okay. Phew. I came really fast on the lead, but okay. So Amir is originally from Egypt. Correct me if I'm wrong. Yes. Yeah. Then he came to Paris to do a PhD. Spent some time in Madison, Wisconsin, and some other places. And then now he's CNRS and part of the Evolution, Genomes, and Behavior and Ecology group in Gif-sur-Yvette. He has a very strong track record studying the mechanisms and genetics of adaptation, balancing selection, morphologies, and taxonomy. And here, his talk is about a former collaborator, Jean-David. Briefly, because we're very ahead of schedule, huge thanks to Virginie for putting all this together. It's wonderful to be in Paris, because like when you're in Paris, you have this mix of memories that come back from your own life, and then stuff you saw in movies, and then you're cycling, and then you see Notre Dame, and crap, that happened a few years ago. Anyhow, thank you for putting together a superb symposium, and we look forward to your talk.
Thanks a lot. So now I don't need to introduce myself, so I will go directly to the talk. But first of all, I would really like to thank Virginie for giving me the opportunity and the honor to talk about the life and work of Jean-David, especially in this great place and in front of great names of our discipline. And I'm particularly pleased that among us there is eminent developmental biologist and Jean-David's beloved wife, Professor Nicole Ledoirin. So many thanks, Nicole, that you are with us. So Jean-David was perhaps the last active member of this generation of French researchers who, in the 70s, during successive discoveries, brought back or helped bringing back Drosophila melanogaster, the emblematic model for genetic study, into the realm of ecology and evolution. Up to the 70s, there was really a real schism in Drosophila research among those focusing on genetics and developmental biology, and then so they are working on Drosophila melanogaster, and those championed by Dobzhansky for sure, working on speciation and adaptation, who prefer to work on other groups like Virilis, Obscura, and Wellistonian, other mostly American and European species groups. So Jean was born in the early 30s in Villeurbanne. Then it was a suburb of Lyon. Now it's part of Lyon. Lyon became bigger. And as a teenager, he developed a very early passion for nature and especially for insects. He has prepared already as a high school student his own collection of beetles that then he will leave it to the Natural History Museum of Lyon. And I remember him telling me, okay, when I asked him why he would then spend 70 years of his academic career only working on Drosophila, so he told me that as a teenager, one of the most influential books that he read, it was by Jean-Henri Fabre. It's a voluminous book. It's 17 volumes, Souvenirs entomologiques. This is really magnum opus of Fabre. And Fabre was a contemporary of Charles Darwin. They shared some correspondences, and Darwin called him an inimitable observer. He didn't have a very strong beliefs in evolution, but yet he was considered as a father of ecophysiology, especially for insects. And like Fabre, Jean was also a very prolific writer. He left more than almost 400 scientific papers, but whereas Fabre worked on a lot of different insects, Jean was very dedicated to Drosophila species, mostly in his research. And this question of ecophysiology, if you, for example, you search for Jean-David in Google Scholar, the most cited paper is called Ecophysiology of Drosophila. So again, it's still the main project that he was interested in early in his career. And the big question of ecophysiology or in evolutionary biology, it has always been what is the interaction between ecology and genetics, as the title of this meeting. But also it's mostly about what is the exact role of the environment in phenotypic diversity. Is the environment a generator of such a diversity, of the evolutionary diversity, like what is now known as the neo-Lamarckian view, or is the environment only a mere selector of genes that underlie these phenotypes, which was by then the neo-Darwinist view of evolution. And perhaps for those of you who have some interest in history of genetics, there was always in France a certain difficulty to accept the neo-Darwinism. It was the mathematical models of population genetics were judged at the time in the 40s and the 50s that a little bit very abstract, and there was more interest in understanding the physiology of the gene, how genes would work, and you cannot do physiology without asking about the milieu or the environment surrounding the genes. And this big debate in the 40s about what is the role of the environment in generating diversity among these researchers that were working. Just to give you a hint, for example, just remember that one of the first major French contribution to molecular biology was the operon system. So again, Jacques Monod and François Jacob, this is a question about how the nutrition can affect the function of genes. And among these French researchers, which were more about what we would call now the neo-Lamarckian view, was Victor Nigon. He was the establisher or the discoverer of Caenorhabditis elegans during the Second World War. And he made his studies in Paris, but then he became professor in Lyon. And Nigon really wanted to know how genes function. And he had established in his laboratory different model systems. And usually he would get different young students and will give different models to different students to try again to ask how they can, the environment affect their phenotypes. And among the models that were in Victor Nigon's lab, so there was the Drosophila melanogaster, and it was for the young Jean David to work on Drosophila melanogaster as a master student. At the time, also, you get recruited, you are hired for a lifetime to make your master and PhD and become professor almost at the same place. So at the time, so Jean was already in his master. He was already hired by University of Lyon. So this is different from how things are now. So, Nigon asked, so they get to have to compare two strains, and the strains that Jean will compare, so he will compare only two strains, wild type and the vestigial. And he will ask for this vestigial strain from Philippe Léritier. And why this is interesting? Because whereas the majority at the time of the French biological community were of the neo-Lamarckian view, very few were really among these neo-Darwinist new ideas. And among them, there was Georges Tessier and Philippe Léritier, who made a postdoc with Thomas Morgan. And they established this very famous experiment in the 30s about population cages, where they showed that natural selection can indeed act directly on genes in the lab, in the laboratory. So they went into the ceiling of a marine biology station where there is a lot of wind, and they put two cages in which they will put two types of flies. One was winged flies, the wild type, and wingless flies because they have the vestigial mutant. And they show that if the cage is closed, so the effect of the strong wing is not very important, the wild type will outcompete with the vestigial. So it will mostly the wild type which will win as a competition. But if you open the cage and you leave it with the strong wind, it's going to be the vestigial mutant which will remain in the cage. And for a lot of naturalists, this is even have been discussed by Darwin, that when you go to windy islands, most insects usually are wingless. So this is a very recurrent theme in evolution. And this was an early demonstration that natural selection can really act to select for otherwise deleterious mutation. So by taking this same vestigial mutation, Jean, during his master's thesis, will ask whether the phenotype will always be the same if we change the environment. And he will change two environmental factors. The first is temperature. And indeed, if you raise these vestigial flies in high temperature, they will develop normal wings. This has already been shown in the 30s. But the second observation, which was very surprising, is that even under the same temperature, if you raise the vestigial flies on a milieu, on a medium that has already been worked out by larvae from the previous generation of vestigial larvae, the flies will develop normal wings. So there is something that will change biochemical constituent of the medium, which would help that this vestigial mutant will no longer be expressed. And this, if you come back to the population cage experiment, would be like, okay, if Philippe Leriti and Georges Tessier and Yvette Nifs would just left for their flies for 2 generations on the same milieu, perhaps this experiment wouldn't have worked. But Jean would never go to say that okay, Tessier at the time, well, yes, there was some, because Tessier was the head of CNRS at the time, and it was a very famous researcher in France. But anyway, so after this observation, Jean decided to conduct his PhD thesis on the effect of nutrition on the phenotypes. And he will study what he called effet prolongé, prolonged effect, is exactly what we call now epigenetic. And he will try different components, asking on the phenotypes if it can be transmitted to next generations. And he will only, among the plethora of elements, only one will give a good result. It will going to be the methylene blue. For just 2 generations, will have an effect on body size or egg size. And following this thesis, he will have a short postdoc in Edinburgh, which was at the time a temple of quantitative genetics headed by Weddington, with a lot of ideas, again, about the role of environment in generating diversity. Back then, Jean will, now he's professor in Lyon, he will found the genetic and experimental entomology laboratory. And I found, and he will get a lot of students, mostly working on the effect of nutrition on the Drosophila quantitative traits. And especially with a certain interest about fecundity and fertility, which are known as life history traits or fitness-related traits. And I found this passage, which I translated from his thesis, saying, okay, yes, fitness is very important for evolution. It's something that we can compare between races, between species, but let's have first, but it's very difficult to measure. Any evolution biologist knows that fitness is very difficult to measure experimentally. And so he will say, let's first start by a very practical goal, very easy goal, just to say what are the ideal medium for each trait, which means that which medium can really produce the higher mean and the lowest variance, phenotypic variance for each of these fitness-related traits. And then the whole research program would mostly focus on this. For almost 10 years, Jean will only worked on 2 strains, vestigial and wild type. And even if himself he has worked on some described new species of beetles and of earwigs, he never worked on Drosophila in the wild. So there was really very separation between lab and ecology at this time. But in the meanwhile, Georges Tessier continued at Gif-sur-Yvette to try to use melanogaster as an evolution model. But this was very difficult for 2 reasons. First of all, most population of Drosophila melanogaster from North America, Europe, and Asia, especially Japan, have had relatively low genetic variation. So to study adaptation, this was not very ideal. And the closest relative to Drosophila melanogaster, which is Drosophila seminos, is also a cosmopolitan species. It's so divergent, so it's not, again, ideally to study the genetics of speciation. But Tessier's student, who was Charles Bouquet, has really keen interest in empirical population genetics. He worked on Crustacea, but he will ask a researcher in his lab, Leonidas Tsakas, who was a systematist, to mostly explore the African diversity, which at the time was not very known. And Tsakas will have 2 brilliant students who will really work on this Afrotropical diversity of Drosophila, Françoise Lemunier, who was a geneticist, and Daniel Lachaise, who was a very brilliant ecologist. And Tsakas would secure a very important grant, and he will invite other researchers to go to different parts of Africa to collect flies and to study those flies. And among those researchers is going to be Jean David for his work in Lyon, and Jean will spend 1 month in Gabon studying flies for the first time in the wild. And this mission is going to be life-changing for Jean. For the first time, he will really recognize how much there is a great diversity of species of Drosophila. It is not only wild type and vestigial. And also, again, he because now he recognized very well the wild type and the vestigial strains, he would realize that he can recognize the difference between an African and the European flies just by some quantitative traits. And this will help then, after this mission. I don't think there is a single continent or a single different part where there can be flies where Jean will not go and collect flies and measure the traits and bring them back to the lab. So in the 70s, he will start really to have this very interest in wild populations and conduct a good collaboration with both Tsakas and Lachaise. And these three researchers together will double the known species from the melanogaster species subgroup. So up to this date, only 3 species were described. 2 are cosmopolitan, melanogaster and simulans, and only in the 50s, a Swiss entomologist, Hans Burla, has described one from Côte d'Ivoire, Drosophila jacoba. And only in the span of 5 years, in different parts of Africa, 4 more species are going to be described. So Erecta, Orena, Tesseri, and Mauritiana. And Mauritiana will be, which in Mauritius would be the first case where you can really make a lot of inter-specific hybridization studies. So the first time that you can also make speciation studies in Drosophila melanogaster. A second important results from this work on African populations was the origin of Drosophila melanogaster. So in a Nature paper published with Charles Bouquet, Jean will compare several populations of Drosophila simulans and Drosophila melanogaster from tropical and temperate regions. And he will show that for some traits, you can really find parallel patterns, not to say adaptations, but for example, always in temperate regions, flies are going to have bigger size than in tropical ones. And this mimic what you would have in the lab if you would put the same flies, isogenic flies, in different temperature, in cold environment, they will always be bigger too. But for other traits like ethanol tolerance, alcohol tolerance, only melanogaster show a strong latitudinal decline, but not simulans. And the explanation at the time, it was that melanogaster arrived earlier to Europe than simulans. This was the hypothesis. And it became strongly adapted to fermentation and vineyard and winery cellars in Europe. So this was a French hypothesis, but I think that it has a part of truth, fundamental truth. And this at the time, there was a big controversy about neutralism and adaptationism in molecular evolution. And almost every new test of selection at the time about how you can test for a signal of selection, it was on the alcohol dehydrogenase gene, because it was one of the rare genes where you can really relate to ecology. So if Jean collaborated with Baghdad and Friedman, and there was a lot of collaboration with Chicago and with other work. Here you see it also with Kimura in Japan. But so now Jean will find himself in his research in the mid of this evolutionary biology controversy. So following the death of Charles Bouquet, Jean will become the third director of the Gif-sur-Yvette laboratory. And at the time, he will no longer work in Lyon, and he will decide that if you want to stay at Gif-sur-Yvette, you should work on Drosophila. So most researchers working in the group, either they will continue to work on Drosophila or they remain. So it was a little bit conflictual, but at least at the end, it gave really a very homogeneous production of the institute, which mostly decided that the whole institute should be dedicated to the study of evolution in Drosophila species with interest in subgroup. And I will just stop on 2 results from this part. It was about the 8th species discovered in the melanogaster subgroup. So this was discovered by British undergrads working on the Seychelles. They found that some species were found only on toxic fruits of noni, which is Morinda citrifolia. It's a plant it's a tropical plant with toxic fruits. And it was recognized as a new species by Gerard Beckley, and then described by Beckley and Sakas as the 8th species, so Drosophila seychellia. So once Jean heard that there was this seychellia on Morinda, he will go to the Seychelles and he will establish a laboratory strain. And this same laboratory strain will be the subject of a lot of different kinds of studies, going from genetics to physiology to reproduction to behavior and finally, and even for molecular evolution and transposable elements. And the whole lab really would be united mostly on a strain. And currently, Drosophila seychellia is one of the best understood model of insect-plant genetic interactions. The second interesting finding coming from this period too, it was about the history of Drosophila melanogaster. So not only that melanogaster comes from Africa, but Jean going to breweries and in bars in Africa would recognize that some flies not look similar to other African melanogaster. And by looking good to them, they looked European. And because European and African flies differ in their allozyme profiles, he will be able to show that if you go in the bar, you are going to find only European flies. And when you go outside the bar, according to how much the distance that you take, the European alleles are going to diminish, and it will be mostly about the African alleles. So, and then if you take the flies that will go to the bar and the flies which remain in the forest, they show some degree of reproductive, pre-cooperative reproductive isolation. So this was not only that melanogaster has evolution history, it has also a current, contemporary speciation history coming back to Africa. How much this would evolve in the future, we cannot know for the moment. So in 1998, Jean would become emeritus researcher, and he will continue working in his lab as emeritus researcher for another 20 years. I think that most of you who knew Jean know him as emeritus researcher, and yes, but he was completely active at this time. And this was an era with strong senses about what he did in Lyon and what he did in Gif. So now he will ask the question not again about what is the role of the environment, but mostly about how this effect of environment, that at the time now, the term that has been used was phenotypic plasticity, how this phenotypic plasticity could genetically differ between populations of the same species or even between species. So how, so it was a kind of a census, no longer about like two opposite hypotheses, but really how selection can formulate the shape of environmental response, phenotypic response to environmental change. And this was done a lot with his PhD student, Patricia Gibert. She is now research director in Lyon. And this is just to give you a hint about this is very famous kind of sheets that were found everywhere where Jean will go, which I will have in his office, in the lab, and a lot of parts. So any single strain which will go enter the lab should have this kind of sheet with different phenotypic traits scored for always 10 females and 10 males, at which date, what is the origin of the species, the strain, and which temperature it is. So there is always 8 temperature, and for most species, and there's a lot of data, even unpublished data about different species, always you can draw what is called the reaction norms about the same phenotype and what are the different values of variance or mean of the same phenotype according to the developmental temperature. And he really showed that these curves have genetic components. So the way how we react to the environmental change itself has a genetic basis and itself can respond to selection. And also, after a while that he did not go back to Africa, so he returned to Africa in the end of the 2000s, first by the gate of Egypt. So that's first how I got contact with Jean. I was a master student, and I found these beautiful flies in desert in Egypt. So I don't know what it was, and then I found that there is someone in France who worked a lot on these flies called Zaprionas. I sent it to him by mail. He answered me that, no, this is not one species. There are 2 tropical species that enter Egypt at the same time, tropical to live in the desert, and that he is now booking his flight tickets to come to Egypt to look at it. And that's how he came, and I knew him, and then I made my PhD in his lab after on this appearance. I also got the privilege after to work a lot with him on, we made several field works in Madagascar and surrounding islands. And because this, in Africa, there are 2 regions which are very less explored, the Congo Basin and most of Madagascar. And in one only mission that I wasn't with him, there was also always his ex-postdoc Vincent Debat with us. So the only mission that I did not participate in, they made a very major discovery. It was on another population of Drosophila from this species called Drosophila jacoba, again on the same toxic fruit. So this adaptation to the toxic noni happened twice in 2 independent lineages in 2 different islands of the Indian Ocean. And, and now I had just a PhD student working on this question. She has just defended a few months ago. Also, Jean worked with Virginie in several missions to Sao Tome, working on with a different project on Brazil evolution on Drosophilids. So the last mission that Jean did on the in Africa, he was 87-year-old, and when he gets sick at just two years after, when I visited him, he was still planning which African country we should go to collect flies from. There was also a common saying in Gif-sur-Yvette, where we are, and where Jean left a lot of strains and different species, that for Jean, every single strain has its own scientific project for 5 years or 15 years to be done. So there are a lot of unfinished projects, and he has a file called All What I Saw and I've Never Published. So and it's really as much as what he's published. So it will take time to go into these files. So I would like to thank all of you, and especially those who helped me to get to learn more about Jean's life. So for sure, Nicole Ludwarin, Bruno David, Jean's nephew and current president of the Museum of Natural History in Paris. Michel Bouletreau, the first PhD student of Jean in the early 60s. I was the last in the 2005, 2008. Yves Carton, Pierre Capi, Patricia, and Vincent, who draw this very nice drawing of Jean. Jean was very proud of this drawing. He put it on his office at Gif-sur-Yvette. Thank you all. Thank you.